All right, so going on to the next part. So we talked about carbohydrates, we talked about lipids, we talked about proteins. Now we're going to move on to lipids. Okay, lipids are fatty, oily, or waxy organic compounds. Now, the biggest characteristic for lipids is that they are hydrophobic. Okay, hydro, water, phobic, fearing, so water fearing. Okay, so because they're hydrophobic, they are insoluble in water. So lipids are kind of a catch-all group for these insoluble in water molecules. Okay, and because they are insoluble in water, that makes them what? It makes them polar, or does it make them nonpolar? It makes them nonpolar. All right, so lipids are kind of a big catch-all group for nonpolar molecules. So you have fats, phospholipids, waxes, and steroids. Okay, and because it's kind of a catch-all group, there are different types of monomers. So whereas in the other three, the carbohydrates, proteins that we talked about, we'll finish with nucleic acids. Those have all three have distinct monomers. Lipids don't. Okay, so you don't have to memorize the monomers for each of these lipid groups. Okay, all right, but the biggest things, lipids are hydrophobic, insoluble, nonpolar. All right, so here you have again those four types of lipids. You have fats and oils, phospholipids, waxes, and then you have sterols and steroids. Okay, and then the functions. All right, so the first group we'll talk about fatty acids. So fatty acids, you have a carboxyl group at one end, and then a long carbon backbone. Okay, all right, and the carbon backbone is made up of carbon and hydrogen. Okay, so hydrocarbon backbone. All right, now if you have the hydrocarbon backbones can be either saturated or unsaturated. If they are saturated, such as this guy here, it means every available carbon has four bonds associated with it. Okay, unsaturated fatty acids. Okay, it means you got some double bonds in there. Okay, here you got one, and this one you have multiple double bonds. Okay, so both of these are unsaturated fatty acids. Triglycerides are the most common fatty acid. Okay, so you have the carboxyl end, and then a three hydrocarbon tail. Okay, so three fatty acid tails. They're most abundant energy source in vertebrates. All right, and we find these fats concentrated in adipose tissue. Right, the difference between oils and fats is kind of the structure of these lipids at room temperature. Oils are liquid lipids at room temperature. Fats are solid lipids at room temperature. Right, again, you may have heard in nutrition this term trans fat, okay, and being kind of really bad. Not good to have a lot of trans fat in your diet. The reason why they are bad because it raises the bad cholesterol in your body. Well, what is a trans fat? Basically, it's it's an unnatural fat, right? That's chemically produced or chemically modified. Okay, so here you have a cis bonded unsaturated fat, basically a trans fat. All right, this is basically moved over to the other side. Okay, and for whatever reason, it makes the fat bad. Okay, the double bond straightens the molecule. Okay, instead of having this sort of kink, it straightens the molecule. And you see that what has a lot of trans fats are a lot of processed foods. Now, the good fats, monounsaturated fats, they help lower the risk of cardiovascular disease. And so that's what you can find in a lot of these things that are recommended to eat. Maybe not in high doses, but have regularly in your diet for their benefits. You also have polyunsaturated fats. Again, things that you know or essential fatty acids can be found in fish. Okay, and so that's why it's recommended to have some fish in your diet for these omega-3 and omega-6 fatty acids. You can also find them in some other products, but again, they have a health benefit, can reduce cardiovascular disease. All right, the next class of lipids are phospholipids. Okay, we'll really talk about phospholipids when we talk about cell membranes in the next lecture. It's the main component of cell membranes. All right, and if you look at the structure of phospholipids, they have kind of two parts of it. They have a hydrophilic head and hydrophobic tails. All right, and it's very important that they have those two parts. All right, it helps them carry out their function. So they have a hydrophilic head and hydrophobic tails. All right, and this allows them to create lipid bilayers. And we'll talk about lipid bilayers and the importance of cell membranes when we talk about cells and membrane transport. But those cell membranes are able to carry out their functions because of these phospholipids. Okay, so in a cell membrane, phospholipids are arranged in two layers called a lipid bilayer. All right, and so you have the hydrophilic heads pointing to the inside of the cell and the exterior environment. All right, and then the hydrophobic tails are in the interior of the cell membrane. All right, so basically you have two layers of phospholipids interacting together, and that's why it's called a lipid bilayer. All right, and those hydrophobic tails are sandwiched between the hydrophilic heads. Okay, so you see here. So let's say this would be the external environment. This would be the cellular internal environment. All right, well, the hydrophilic heads are facing towards the outside and towards the inside of the cell. All right, and then the hydrophobic tails are facing each other. Okay, and it's really important to have this structure because it, like I said, it helps the cell membrane carry out its function. All right, so the next group of lipids are waxes. Basically, it's just a mixture of lipids and long fatty acid tails bonded together to long chain alcohols or carbon rings. They're water repellent. All right, the molecules are really packed tightly together, and they're nonpolar. Okay, and so they don't readily dissolve in water, so it makes these wax molecules water repellent. And we see a lot of waxes produced in plants because it helps them to restrict water loss. And then the last group, steroids. These are lipids with no fatty acid tails. They play a major role in cell membranes. You have cholesterol, which is a steroid. You have sex hormones, which are steroids. Okay, so there are major biological functions of these steroid lipids. Okay. Don't worry about LDL and HDL. That's just two different types of cholesterol. HDL is good, healthy. LDL is unhealthy cholesterol. And then you have, again, testosterone, estrogen. Two of the major sex hormones are examples of steroids. Okay, takes us to the last major group of organic macromolecules, nucleic acids. So we talked about carbohydrates, we've talked about proteins, we talked about lipids, and now we're finishing on nucleic acids. Okay, so what are the monomers of nucleic acids? That's the nucleotide. All right, the nucleotide has three components. Okay, so here is the basic structure of a nucleotide. You have a pentose sugar. Okay, what do you think pentose means? Pentose means it's a 5-carbon sugar. All right, that 5-carbon sugar can be either deoxyribose or ribose. You have nitrogenous base, and there are 5 types of nitrogenous bases. And then you have a phosphate group, okay, or 1, 2, or 3 phosphate groups. Okay, all right, and so this is the basic structure of a nucleotide. All right, so here are the nitrogenous bases. So you have your adenine and guanine, okay, your purines, and then you have thymine, cytosine, and uracil, your pyrimidines, okay? So based on the structure, what's the major difference between purines and pyrimidines? Purines are double-ringed nitrogenous bases. Pyrimidines are single-ring nitrogenous bases, okay? All right. And then let's see. So that what we see here, the adenine, guanine, cytosine, thymine, and uracil, that's what would go. That's what would go here. Okay. Then you'd have either ribose or deoxyribose right here, and then your phosphate group or groups right here. All right. Let's look at the structure of ribose and deoxyribose. What's the big difference here? Okay. We know they're both sugars because it ends in OSE, remember? And they're both 5-carbon sugars because they're pentose. All right. Sugars. All right. So what's the difference between ribose and deoxyribose? We'll look at the name, deoxyribose, meaning that it has one less oxygen. So there's the oxygen in ribose that is not there in deoxyribose. All right. ATP is a very, very important nucleic acid. Okay. It stands for adenosine triphosphate. All right. So you have an adenine nitrogenous base. You have your pentose ribose sugar. Okay. And then you have 3 phosphate groups, triphosphate. And as we'll see a lot, not only in this class, in other courses, you'll come back to ATP. You'll see ATP over and over again because it's an important energy carrier molecule in organisms. Okay. All right. So we'll come back to that when we talk about metabolism. All right, so what are some differences between DNA and RNA? Okay, so DNA is deoxyribonucleic acid, RNA is ribonucleic acid. Those are the 2 major types of nucleic acids. Okay, all right, and so the first difference is you have deoxyribose and you have ribose. Okay, so the pentose sugar is different between the 2 types of nucleic acids. Deoxyribose in DNA, ribose in RNA. All right. Another difference is nitrogenous bases are different. In DNA, you have thymine, adenine, cytosine, and guanine. Okay. And adenine and thymine always bond together. Cytosine and guanine always bond together. In RNA, you don't have thymine, you have uracil. So uracil and adenine bond together. Then you have cytosine and guanine bonding together. Okay, so that's another difference. Thymine in DNA, uracil in RNA. And then the third major difference is DNA usually exists as a double-stranded molecule. So here's one strand of DNA and another strand of DNA bonded together to make a complete molecule of DNA, so it's double-stranded, whereas RNA usually exists as single-stranded. So 3 major differences between DNA and RNA. Okay, difference in pentose sugar, deoxyribose versus ribose. Difference in nitrogenous bases, thymine in DNA, uracil in RNA. And then the overall structure, you have double-stranded molecule in DNA and a single-stranded molecule in RNA. All right, so that ends this lecture. Major things you need to know. Again, what are the 4 types of organic macromolecules and their defining characteristics? What are their monomers? Okay, what's the difference between condensation and hydrolysis reactions, and what is overall produced in those reactions? Okay, again, this isn't a exhaustive list of things you need to know, but if you can answer these questions, all right, you are building a good foundation from this lecture. Okay, as always, let me know if you have questions.